Hello everybody, this is going to be chapter 4 on microscopy, staining, and classification. One of the first things that this chapter talks about is the metric units of measurement, and of course this is used in conjunction with things like microscopy to try to understand how small or the scale of things when compared to others. So make sure that you're able to go from one unit to another. So microscopy in general, its purpose, of course, is to magnify and to see things that are unable to be seen with the naked eye. And so there's different principles that we need to understand as they relate to their function. The first being wavelength. The electromagnetic spectrum, for example, we see it as waves, and these waves are essentially increasing in frequency from the right to the left, with the lower energy waves going towards the right and the stronger waves going towards the left. Of course, gamma rays being the strongest, infrared, microwave, and radio waves being the weakest. And here we have the different properties of light and convex glasses. So here you see a specimen. As light is reflected from that specimen, it changes the direction of light. This is known as refraction. And so as these beams of light travel, at some point there's an intersection between them, and that's going to be our focal point. And this principle is what's used for a lot of different types of light microscopes, and to some extent electron microscopes as well. So resolution is just the ability to distinguish two points that are close together. How good they're being able to be seen, the better the resolution, the better you can distinguish one object from the next. And different microscopes have different powers, of course. And here we can see kind of the scale of everything with atoms and some really, really small particles like viruses being in the nanometers. And then you see typical bacteria now starting at micrometers, with eukaryotic cells getting closer to the 10 micrometer mark. And then you have some larger organisms, of course, as you get closer to the right. And here below this figure, you can actually see which microscope would be appropriate to examine these types of organisms. So contrast is just the difference in the intensity of light, and a lot of these different microscopes use that system. We even use that system in our cell phones, for example. A lot of the focusing happens via contrast-based systems, and so it's just the difference between the lightness and the darkness of the colors or the objects that you're seeing. And this is really important, of course, to be able to distinguish the difference between what is one object and what is another. Whenever you stain things, however, it makes it just a little bit easier to identify those objects that are in the foreground and those that are in the background. Bright field microscopes, these are the simple ones that we all know and love. They have magnification, and you can see basically by exposing these objects or specimen to light. The foreground is the object, the background is light. And these could be compounds, so they have a series of lenses. Typically the light passes through the specimen and you're able to see it. One of the things about this type of microscope, though, is that it sometimes requires oil, and we examine these specimen or samples with or under oil immersion, as it's one way to increase their resolution without having a lot of distortion in the image. Of course, total magnification is just calculated by looking at the eyepiece and the objective lens or the ocular lens. And most of these have a condenser light under, so that you're able to modify how much light goes in, contrast, and all that other stuff. So here you can see a very, very basic microscope where you have the ocular lenses on top, you have the objective lenses or the revolving nose piece where they're organized, and then you see the light source and all those other components. On the right, you can actually see how the line of vision is impacted by the different elements within the scope. And here we have a slide just talking about oil immersion, and light does a very interesting thing, or the medium that it's passing through does something interesting, and that's to refract light. And so this refracted light sometimes messes with the image quality, with the contrast, with your ability to see things clearly. And so when we prepare a slide with oil immersion, we add a drop of oil, typically some type of mineral oil, and we make contact with the objective or the microscope's objective. And so by doing so, we can focus light a lot better, minimizing that refraction so that we're able to see a lot better, because at this magnification, typically we're talking about 1,000 times magnification or greater, you start having interference even from the air that's in between your specimen and the microscope's objective. Now, dark field microscopes, they're really good for pale objects. Typically, you're going to have a dark background in this case compared to the light background that you saw in the ones before. And this is for things that are small and colorless. And then you have your phase microscopes. So these phase microscopes are essentially consisting of two different lights. So you have phases that are created by these lights, and depending on how out of sync they are or how different they are, differences in the light passing through, you're going to create contrast in an image. And the same is true for differential interference contrast. And we'll see some images right here so that you can kind of examine what they look like. So on the very, very top, we have a bright light or bright field microscope. As you can see, there's very little that you can see there because there's not enough contrast. You can see very simple outlines. And then if you were looking at a dark field, you can see some of the bacterium, but you really can't see the shape of the cell. Whereas with those phase microscopes, you can actually see those quite much, a lot better actually. Another type of light microscopy is the fluorescent microscope. So this is where a UV light, in this case, illuminates the object, and the specimen or the objects that you're trying to look at sometimes can radiate that energy back. And in this case, you're going to see some fluorescence. Some of them are naturally fluorescent compounds. Others are fluorescent dyes that are added. Sometimes you can even attach these fluorescent dyes to immunoglobulins or other proteins so that you're able to tag specific organisms with it and illuminate those as a consequence. So here we can kind of see some of that fluorescence in action. And so here we have that immunofluorescence. Of course, we have that fluorescent dye being attached to the antibodies, and as those antibodies are attaching to the bacterium, then you can kind of visualize them with that fluorescence. Now, confocal microscopes, they still use fluorescent dyes, but this one uses UV lasers to give that fluorescence to the chemicals, and you're able to kind of build up layers of these single planes to create a three-dimensional image and then digitalize it so that you're able to see a very interesting picture of that. Now, electron microscopes, again, we start now looking at things at 10,000 to 100,000 times in magnification. And these are very, very tiny particles that we're looking at. We're looking at viruses, bacteria, components of cells on the inside. And you have two types, your transmission electron microscopes and your scanning electron microscopes. So here we can see the differences between a transmission electron microscope and a traditional light microscope. As you can see, still we have beams of these particles. Again, in this case, it's not photons, but electrons. And then, as you can see, they're eventually seen at the final stage after being refracted through these different components. And here we have several different scanning electron microscope images. These are a little bit more for visualizing the surfaces and the features, the external features of microorganisms, as you can see right here. So they create kind of this neat three-dimensional image of what these look like. With probe microscopy, these are things that are now magnified 100 million times. And you have scanning tunneling microscopes and atomic force microscopes. On the left, we have the scanning. As you can see, they're meant to visualize the surfaces of objects that are really, really small. And on the right, with that type of microscope, you can see individual atoms, molecules. You're looking at things that are extremely, extremely small. Table 4.2 has the comparisons of all these different scopes. Please make sure you review that in your text, as it has some valuable information, including the features, the description of the image, and what they would typically be used for. Now, staining is a very important part of microbiology because this is one of the ways we can color specimens and we can distinguish between them. Not only that, but also visualize their shapes, their organizations. And so you have different procedures for these. So here we have an example of fixing or preparing a sample for for staining. So you spread the culture over a slide, then you let it dry, typically so that there's no moisture. And then after all the moisture is gone, you can pass it through a flame to fix it or attach it firmly to the slide so that when you are going through the process of staining, none of these bacterium or microorganisms are lost. When it comes to dyes, most of the dyes that we use to visualize some of these microorganisms are salts. The chromophore is the pigment or the colored portion of the dye, and you have some acidic and some basic dyes. Typically, acidic dyes are going to stain any basic or alkaline structures, and basic dyes are going to stain the acidic structures. So simple stains they're fairly simple dyes. You have things like crystal violet, safranin, methylene blue, things that are used to visualize things almost immediately. These are for the shape, size, arrangement of your cells. And here you can kind of see some bacteria without a stain and some with a stain. As you can see, they're much better visualized by the utilization of that stain. You also have differential stains. So the gram stain, for example, is one of the most common differential stains. So typically you're going to prepare the slide for staining. Then after that, you're going to add your crystal violet. After crystal violet, you're going to use iodine or grams iodine solution. What this is going to do is act as a mordant. And so this is going to stick to the inside of some of the cells. It's going to make it thicker, more difficult to be removed. And then you use a decolorizing solution to remove any of this dye that was not stuck to the inside of cells. And so as a counter stain, after all your non-attached pigment is removed, safranin acts as a counter stain so that you're able to see the differential stain in this case. So anything that used that iodine as a mordant and a lot of it gets stuck inside the cell, that stuff is all going to look purple, and everything that was not stuck inside the cell is going to look pink from the safranin. And this is one of the techniques that we used to differentiate between the gram-positive and gram-negative bacterium. Of course, we have several different stain types, which we'll talk about briefly. But here you can kind of see the gram staining procedure. Of course, this is outlined in the text as well, so I encourage you to visit that. So here we have another example of a differential stain. So there are some bacterial species, like the bacteria that causes tuberculosis, that are a little bit different to other bacterium. So in this case, this specific bacterial type, Mycobacterium tuberculosis, has a lot of lipid present in the cell wall. And so whenever you try to do a traditional gram stain, it's difficult for these stains to penetrate. However, with acid-fast bacteria, you can use different stains, and these stains are going to stain that that cell wall a little bit better. Then you're going to rinse them again with a decolorizer, and then you're going to use methylene blue as a counter stain. So it looks a little bit similar, as the colors pink and purple are really similar to red and blue. And but again, this is for a different type of bacteria, the acid-fast bacteria that we call them. Here we have an endospore stain where I think this is malachite green that stains the endospores, and safranin staining the rest of the coat. We also have other stains like the GMS stain. So the GMS stain is really good at staining carbohydrates, polysaccharides in general. And the HE stains, the hematoxylin and eosin stains, these are really good for looking at tissues, human tissues to be more specific. It's one of the ways that we can actually visualize cancer cells and other types of components that are changing in tissues. The hematoxylin stain, this one's going to stain things purple, if I'm not mistaken, and the eosin is going to stain them pink or reddish in color. Then we also have some special stains. Negative stains, these are the ones that are going to stick to typically negatively charged molecules. In the case of bacteria, you're going to see these are fairly common to identify capsules. So these are also known as capsule stains. And flagellar stains are also important, just so they can tell us a little bit about how many flagella they have, their organization. And typically you have things like paranosinylene, which is going to use a mordant to kind of bind to this flagella. And as they bind to that, they're also going to increase the diameter of the flagella, allowing us to visualize them, again, because they're typically really, really thin. So you can see that negative stain, or the capsule stain, and then here you can see the flagella visualized. Table 4.3 has the type of stains, their results, and why we would ever look at them. So make sure to examine those as well in your text. Now, electron microscope staining is a little bit differently. Because you're dealing with electrons, you need something that's able to capture them, and so heavy metals in this case would be appropriate for that. So these stains are going to be much better visualized when you have the presence of some of those heavy metals in your specimen. Now moving on to classification. So classification of organisms, that's known as taxonomy, and this is where we organize living things based on the information that we have about them. It helps us understand the relationships. It helps us understand similarities and differences between the organisms that we have. And so in 1753, Carolus Linnaeus established a system where he used binomial nomenclature, and using the two-name system, which is the binomial nomenclature, we were able to describe individual organisms almost as if they were to have a first and last name. Where the genus and the specific epithet would be kind of the culmination of the levels of organization, where things that were capable of interbreeding were its own genus and species. And so, of course, the more we know and we understand about life and the characteristics of life, how we define species, which are still things that are interesting to learn about, as we don't have concrete definitions for some of these things, they really help us understand these groups and how we organize them. And so here we have a basic layout of the levels in a traditional taxonomic scheme. I'm not sure if you all remember from when you were in grade school when they talked to you about classification, where they said that King Philip came over for good spaghetti. Of course, that's lacking domains, but for the rest of them, it works pretty well. So you have kingdom, phylum, class, order, family, genus, and species, of course with domain at the very, very top. But it's a great way to classify living things, as it kind of breaks them down by similarities. So originally there were supposed to be only 2 kingdoms, but the features of these organisms, as we examine them further, you had 5, and then you had 50, and then you had 30, and then you even had changes, right? So at some point, fungi were classified within the same branch as plants, and of course now we know that they were different, so now we classify them into their own groups. But again, the goal of this was just to classify, catalog, and make sure that it reflected the phylogenetic hierarchy. So the organization of this was phylogenetic in nature. You were able to trace back lineages. And domains were established after we were able to sequence RNA subunits, and after doing that, the scientists that figured this out were looking at those ribosomal subunits and found that, well, they were different. And so based on those differences, they were classified as 3 larger domains: eukarya, bacteria, and archaea. And so that's still what we have now. Of course, the more we understand microorganisms, the more things change. But as of now, this is kind of what we have. There are different things that we use to classify, including physical characteristics and a variety of tests. The first is physical characteristics. There are characteristics that are very unique to some of the morphological characteristic of species, and so we use that to classify some of them. There are very, very clear appearances for specific types of bacterium as well, so we use those. Then we have the biochemical tests, and so these can range from the production of chemicals within a test tube, to even using selective and differential media to try to see if an organism is lactose fermenting, for example. So here we have one example of different types of bacteria. And so as you are culturing them, you might notice that there are some that are producing bubbles as they're fermenting some sort of chemical compound. On the right, you see some that are capable of producing hydrogen sulfide as a byproduct. And in the presence of iron, it would form a dark precipitate, and you can kind of see that from that biochemical test. There are different assays that we run And by doing so, we can kind of detect different species by looking at the specific colors that they produce. Now, serology is a very interesting field. What they use in these serological tests to identify bacteria is they use specific antigens, and these antigens are either going to agglutinate or not agglutinate within a sample. And what that's going to do is going to form those little clumps after the particles are opsonized. So they're surrounded by those proteins, eventually leading to the little clumps and clusters. So this is really good because if you know that a specific antibody is working on that specific microorganism, then you can identify it positively. Phage typing is also really interesting because bacteriophages are highly specific to the bacteria they infect. You can set up these beds of bacteria, also known as the bacteriolons, and you can add a droplet of some of those phages. And what that's going to do is going to develop into plaques. So plaques are essentially zones of inhibition caused by those bacteriophages. So the bacteria, in this case, is killed by the bacteriophages, leaving that empty space there, or that area where there's no growth. Then we have other tests like the Mal-Dietorf mass spectrometry. This is when you use mass spectrometry to examine some of the products that are being produced by some of these microorganisms. It's a really good test. It gives you some very, very specific proteins that are being made by the microorganisms, and they're able to give you even information about the ability of these microbes to resist specific types of antibiotics. So this is a really good diagnostic tool. And then, of course, we have the analysis of nucleic acids. That we can sequence fragments, we can look for markers for specific codes, but we're also able to identify the ratios of things like CG content, which is really a valuable tool for identifying specific species of bacteria. And last but definitely not least are the dichotomous keys. So dichotomous keys are typically yes/no responses. And so you see this in even some aspects of popular media, when you're asking like 21 questions, or you go on one of those apps or websites and they're asking you, can we figure out who your favorite superhero is based on these questions? And then, do they have glasses? Yes or no? And then, do they have a cape? Yes or no? Are they from this planet? Yes or no? And so that dichotomy, the yes, no, the present, absent, the 3 or 6 flagella, whatever the characteristic may be, that's going to determine the direction in which the statements will proceed. And it looks something like this. So here it does have gram-positive cells. Yes, no. If yes, it's a gram-positive bacteria. If no, then you have to re-examine where this trail goes next. And so this is kind of how those dichotomous keys are used, not only for identifying microorganisms, but for identifying a lot of other organisms as well. And that is it for this lecture.